All right, hello everyone, I'm Huang. I'm a postdoc at Yale University working with Dr. Vanessa Izenhua. And in the following roughly 15 minutes, you are going to hear me talking about the variation in helminth-induced feeding changes in herbivores and how those changes interact with other trophic levels. So let's start with the food chain. And in this food chain, because we are disease ecologists, and we prefer to see a food chain with parasites involved. So in this food chain, there are producers and then herbivores, which are hosts, and also parasites, which infect hosts. And in this food chain, there are top-down effects of parasites on herbivores, and also top-down effects of herbivores on producers. Normally, disease ecology study focus on the interaction between parasites and the hosts. But because once parasites change something in the herbivores, herbivores can in turn affect producers. So there are indirect effects of parasites on producers through changes in herbivores. And based on different mechanisms, there are two types of these indirect effects. And the first type is density mediated, which means that parasites change herbivore population density and in turn affect producers. And the most famous example is this one. So in Serengeti in the 1960s, rinderpest was eradicated and then wildebeest population escalated. And because they eat grasses, so there was a lower grass biomass. And the second type is trait-mediated effect. So parasites change herbivore behavioral or physiological traits, and then in turn affect producers. And for this type, several experiments show that parasites induced anorexia can cause high producer biomass in aqua systems. However, for terrestrial systems, there was rarely a study. And this model published last year showed that with nematode infection, then reindeer had lower had anorexia and a reduced resource intake. As a result, there was higher producer biomass. And this model showed that this effect could be as strong as density mediated effect, even though there was no mortality caused by the nematodes. However, for this model and also other studies investigating helminth-induced feeding changes in herbivores, they always focus on the average changes. And now we know generally the average changes are negative, which are anorexia effects. However, no study has yet investigated the variation among individuals. And as said by a great disease ecologist just 2 days ago, so now it's the time for us to embrace variation. So per his request, this study investigated the individual variation in helminth-induced feeding changes and and also evaluating the potential causes. And with that variation, how does it affect other trophic levels, including plants? So this data collection was done in Empire Research Center in Kenya, and the study species was Grand Scazio, which is a small herbivore ungulate species. And we captured 9 individuals, and because they were in the wild, so there was helminth infection. So we gave them anthelmintic drugs killing those helminths, and then release them back into the wild. And since they started to forage, helminths started to reaccumulate. And then we used their fecal egg count to be an index of infection intensity, and did behavioral observation, recording how much time they spend feeding. And then we use the foraging time and the fecal egg count, the infection intensity, to evaluate the helminth-induced feeding changes. And this is the result. So X is infection intensity, Y is proportion of time spent feeding, and different colors are different individuals. The black dashed line is population mean. So we can see that for different individuals, they could have different slopes. And if the slope was negative, it means that with the increase of infection, then feeding time decreased, which was anorexia. So if we just take the slope, and then we can see at their confidence intervals, then we can see that the population mean effect was slightly negative. And for the 9 individuals, there was individual variation. And the mean effect could range from slightly positive to 0 to really negative. So for some individuals, they experienced strong anorexia, but for some, there was probably no effect. So the next question was why we observe this individual variation in the feeding changes. So to answer that, we evaluated variables related to the environment, predation risk, or other factors by estimating the tree cover from those locations where gazelles were spotted. And at the same time, tree cover is also a proxy of predation risk for these gazelles. We also estimated the standard deviation of tree cover. Oh, by the way, the tree cover was estimated from remote sensing data. And the standard deviation of tree cover is also heterogeneity in the habitat selection they use, or how broad the spectrum of the habitats they use. So we found significant effects of interaction between infection intensity and heterogeneity in habitat selection on feeding time. And we can see that this plot is totally the same as the previous one, but now different colors are different heterogeneity, different habitat selection strategies. And with low heterogeneity, which means that animals tended to use roughly the same type of habitats across time, then the mean effect was 0 to slightly positive, and with a wide variation. So sometimes it could be negative, sometimes it could be positive. However, with broader spectrums of habitat use, like the darkest color, then there was stronger anorexia. So this could be because animals, if they experience like high predation pressure, they might use broad spectrum of habitats, and then so they have some refuges, but at the same time, they avoid those high predation risk and high foraging quality areas. And in turn, they had lower foraging efficiency. It could also be because using broad spectrum of habitats require higher energy input, or plants communities vary with habitats. But so these are our possible, like best possible explanation for the mechanisms, but it definitely requires more further study. So next, now we know there was variation, and the variation was potentially caused by the environment. We want to know how this variation affected other trophic levels. To answer this, to do this, we couldn't really use the empirical data. So we built an individual-based model, and with consistent number of herbivores. Each herbivore can have different parameter values. And so herbivores can be infected with helminths, and helminths can be shedded into the environment through feces. And once the parasites in the environment were ingested, they could become infecting. And for both parasites within the host and in the environment, they both had death rates with different rates. And in this model, there were also plants, and the plant biomass increased with logistic growth with carrying capacity, and it decreased with herbivore consumption. The last parameter in this model was feeding rate. The feeding rate was affected by helminth infection intensity and habitat selection strategy based on the statistical model I just showed. And once feeding rate changed, it also affected the parasites ingested and also plants consumption. So we did a simulation with 2 steps. The first step was to compare presence and absence of feeding changes and how that affected other trophic levels. So we simulated different proportions of individuals showing feeding changes ranging from 0 to 100%. So for example, 25% means a quarter of herbivore individuals showing feeding changes after infection, but the other 3/4 did not. And for habitat selection and infection intensity, we simulated following the empirical data. And these are the results. So X is the 0% ranging to 100%. And so on the left is feeding rate. We can see that with the presence of feeding changes, then the overall feeding time decreased, because most of the time the feeding changes were negative. And because of a decreased feeding rate, so there was a smaller amount of consumptions of plants. So at the same time, with feeding changes, then there was higher environmental plant biomass compared to 0%, which was absence of feeding changes. The plant biomass could increase up to higher than 10%. And this shows a top-down effect of the herbivore feeding changes on plants. And also because of a lower feeding rate, there was also smaller amount of parasites ingested. So there was also lower infection intensity, and this showed a bottom-up effect of herbivore feeding changes on parasites. And this is the last result for this study. So the second step for the simulation, we use 100% of individuals showing feeding changes. But now we tested different contexts. So now there were different degrees in the habitat selection heterogeneity and infection intensities, ranging from low to high. And these are the results for feeding rate and plants. So we can see, obviously, when there was high infection intensity and the low heterogeneity, there was much wider ranges. And that was because when the heterogeneity was low, the variation in the feeding changes could be larger, and so sometimes negative, sometimes positive. And with the increase of the infection intensity, the variation became even larger. And as a result, we see this wide variation in the outcomes. And if we reduce the plant carrying capacity slightly, the plant populations could totally crash. So in conclusion, we observed this individual variation in helminth-induced feeding changes, potentially caused by the environment or other factors. And this variation had a broad impacts in a community with top-down effects on producers and bottom-up effects on parasite infection. Also, with this variation, trophic interactions might be more complex than previous experiments or model predicted. And due to the complexity, in some circumstances, the variation lead to instability in a community or an ecosystem. So, but now just because we just use a model for testing this idea, and we are seeking collaboration to test these trophic interactions using empirical data. So now it's the end. So we thank people and the field team, and also members in the Izawa lab, and also the funding sources. And I personally thank EID committee and staff for organizing this meeting. And thank you for staying this late to listen to my talk. So I will stay around slightly longer after the conference. So if you want to discuss anything, yeah, you can feel free to come to me and And yeah, contact me in person or virtually. So with this gazelle butt, I'm happy to take any question.
I think we have time for a question while we do the setup.
Hi, I'm over here. I have a question. So that was a great talk. Thanks. I would imagine that spatial heterogeneity and forage quality could also have important ramifications.
Can you be louder? Sorry.
Hello, can you hear me now? Excellent. So I was asking about spatial heterogeneity and forage quality. How would that maybe affect like host condition, parasite fitness, the phenotype, the individual variation in this phenotype? Or do you suspect that there are other drivers in this system that are more important?
You mean the heterogeneity in their habitat selection or spatial heterogeneity?
Yeah, so I mean, you had that figure where, you have
Yeah.
organism, individuals using different habitats. I would imagine that those habitats could also vary in forage quality.
Yes, yes.
So I just
So
was-
there can be a lot of different mechanisms, and so far we can just postulate or hypothesize potential mechanisms, and we cannot really rule out anything. Like predation might be a factor, foraging quality might be a factor, and also like energy use. So everything is possible, and that's why I say like we probably need like further investigations.